Mechanical and chemical digestion begin in the mouth. Mechanical digestion is the breakdown of food, like mastication. In order to allow for easier swallowing, the oral cavity secretes mucin from the salivary glands. Saliva also contains salivary amylase, which hydrolyzes starch and glycogen, producing shorter polysaccharides or maltose. Once the food is chewed, the tough momentum proceeds into a ball, also called a bolus, and pushes it into the pharynx. To prevent food from going into the trachea, the opening glottis is blocked by the epiglottis. The esophageal smoothly relaxes and allows food in the stomach. After the esophagus moves down the food by involuntary contraction of the muscles, also known as peristalsis. Food is then taken into the stomach, where it secretes gastric juice which moves through the food. The gastric juice components have a low pH of HCl, which dissolves into cellular material, but also contains pepsin, which are the chief cells found in the gastric pitch, which combines with the acid and secreted from the parietal cells and activates pepsinogen, the inactivated version called a zymogen. As a result of the mixing between the food and the acid, a mixture called chyme is produced. Springs are found in the stomach, the cardiac orifice, and the pyloric sphincter, which regulates the passage of food and chyme to the small intestine, where most digestion and absorption occur. 25 centimeters of the small intestine is called the duodenum, where most of the chyme mixes with the digestive juices. The pancreas produces hydrolytic enzymes and sodium bicarbonate, which is alkaline. The liver produces bile, which is stored in the gallbladder and contains bile salts. The sodium bicarbonate makes the pH restored at higher levels. Pancreatic amylase turns from polysaccharides into maltose and disaccharides. Disaccharides like maltase and lactase convert disaccharides into monosaccharides. These food particles are then absorbed. Protein digestion begins with pepsin. Other enzymes in the duodenum dismantle polypeptides like trypsin, which breaks proteins into shorter polypeptides, and polypeptides into smaller polypeptides, as well as chymotrypsin. Dipeptidases like carboxypeptidase and aminopeptidase trigger the breakdown of smaller amino acids into amino acids. These are triggered by enteropeptidase. Nucleases hydrolyze DNA or RNA into nucleotides. To digest fats, biostats are secreted to prevent the lipids from coalescing, since fat is insoluble in water. Fat globules are then emulsified. Lipase hydrolyzes fat into glycerol and fatty acids. The rest of the small intestine, like the ileum, function to absorb water and nutrients. The small intestine has macrovilli and villi that increase surface area to aid in absorption. The food particle has now been broken down into nutrients that are transported across the epithelial membrane and throughout the lacteals through active or passive transport. The breast border cells also help to increase the surface area. The intestine connects to the small intestines at a T-shaped junction. The C-stem is the pouch at the junction and has a dispensable extension called the appendix. No digestion occurs here. The large intestine reclaims water. The waste, also called feces, are pushed along the tract with peristalsis and carried to the rectum, where the feces are then exposed to the anus.